I'd like to invite the speakers up to the podium for the Q&A session. And anybody who has a question, please come to the mic, identify yourself, your institution, and we'll be happy to take any questions or comments.
Thank you. David, you go first.
Dr. Denning.
David Denning, Manchester. So thank you, Pete, for that, Pedro, for that lovely talk. I think there's something missing, and that's the problems of chronic and allergic aspergillosis. I realize that doing a randomized trial of chronic pulmonary aspergillosis is difficult because it takes a long time and the outcomes are not clear, but I do think a registry might be a good place to start. The awareness in this country of chronic pulmonary aspergillosis is dreadful. I just did a little quick literature survey. I think there are 3 papers from the whole of the US. We've been describing it for 20 years. There's papers from every other part of the world. This country's, the respiratory community particularly, doesn't seem to get it, I think, yet.
Yep.
In our work in Manchester, we found that the rate of resistance, antifungal resistance in these patients is pretty high, that somewhere between 8 and 10% on itraconazole get resistance, 5% on VORI get resistance, so there's a resistance piece as well. Diagnosis is tricky. There's lots of interesting radiology to be done. There's a surgical interface. I think it would be something that the MSG should do. Globally, we've just been trying to re-estimate how many patients there are. In India, we think there are 1.5 million people with CPA and about 200,000 deaths every year. So globally, we think it's a big problem. It's probably not so much a big problem here because you have less TB, but you do have NTMs, you do have sarcoid, you have patients who have pneumothorax, et cetera, et cetera. So I think this is missing from your schedule, and I would start with the registry, I think.
Okay. It's a wonderful idea. I mean, I think that most investigators or most clinicians in the US will say, even those of us who are aware of it, still don't, at least we don't recognize it. If it's there and we're blind to it, I can't speak to that. But for myself, I don't see a lot of this. And yet, I mean, we see it, we see some. A registry, this would be perfect for a registry. Your point is well taken. This would be perfect for a registry.
John?
Does this work? Yeah, okay. I have a question for all 4 of you. I'll start with Jerry first. I'm always interested in this, and it's for the young people. We talk stats. We talk statistical stuff and all the big data sets and various things up there and say, this is really facile. We can really do this. Where should the MSG be in statistical support for young investigators that want to do these ideas? Because to me, there's 2 issues, cost and cost. First, the cost of statistical support. This is great. Everybody wants the statisticians. All the young people want to try to get them, then they can't get them. They get put to the back of the line and they don't do what they want and all that stuff. So there has to be a time when mycology has statistical support. And is there enough there that you could train this next generation to do enough statistical support to do these databases? And then the second thing is these databases keep getting bigger and bigger and bigger, and they cost more and more to actually access. Should the MSG be available to actually go in there and get those databases? Or is it way above our ability to actually do, and in fact, it's got to be done by industry?
So on the cost issue, I'm going to work backwards. So the database that I talked about, they range from free to very expensive. MarketScan, for example, for a year's worth of MarketScan data, costs about $500,000, well beyond the cost of any one individual. And along that same continuum is the expertise that's needed. Some of these databases, you need very minimal statistical expertise. And if we go into MarketScan or various EDW data, you need extensive statistical expertise. That said, as I mentioned, I already own all these databases. I would be happy to make them as a resource to the MSG. In terms of the statistical issue, and how we provide support for investigators. About a year ago, Pete's wife made my family some spanakopita. So I work for food. It was very good. We still talk about it.
And it's the best spanakopita in the world.
Yeah, it was stellar. It really was. So there are two issues there. While I don't have an infinite amount of time, I would be more than happy to, as Pete had in his slide, I think you said call Jerry. I think that's the payback for me calling you out. Right. On your data collection techniques. I'd be more than happy to entertain requests for people for statistical analysis. In terms of training, the thing that immediately comes to mind, and just off the top of my head, it would be, I think, fairly reasonable to maybe carve out some time at one of these meetings to have a session that's focused on statistical skills, be it basic statistical skills, advanced statistical skills, whatever the level is that is in the room. And we could do all three if necessary to both raise the level of expertise or at minimum raise the level of knowledge, know when you've reached your limit and when you need to call a statistician. So again, I'm more than happy to make myself available. I'm more than happy to make these data sources available. There's no sense recreating the wheel. I've already paid for them. So there's no sense that we shouldn't make use of them as part of this group.
So there's a lot of young people here, et cetera, and old people. You've made a very kind, maybe gift to us or for the MSG to actually, potentially we have an idea. Pete said an idea. Maybe be able to go through the MSG, go through you and stuff like that to ensure it.
Yeah, absolutely. I mean, there would need to be some sort of mechanism. Right. My inbox only holds so much. But as Pete, and our experience, it's a true statement. Pete will email me a question. I'll address it with some data, and he'll decide whether he's going to pursue it or not. Yeah. Is there enough data? Is it viable? And I think that sort of mechanism on a larger scale could easily be worked out.
I think all that, there needs to be something set up to actually be able to do it so that you're not overwhelmed, the system's not overwhelmed, but we shouldn't pay attention to that.
But the other piece of this is that when we embrace a funded project, I mean, we do a lot of non-funded mandates. MSG 19, which you'll hear about tonight, is a non-funded project, and Jerry has donated his time for that. But for other funded projects, we have a line item for biostatistical support. If we design a trial, if we get involved as helping in trial design or sit on the DRC, there's a line item for biostatistical support. So that's where that comes from.
Okay, I don't want to take all the time, but I had 4 questions, and I'll go to the next person. So, Dr. Nguyen, I think you're actually right. One of the things we don't do a good job of, I always believed that one species or one strain caused infection. You suggested that maybe there's more than one. And that's just the way that we do the SOPs or our cultures, and now we got sequencing and stuff like that. So I would emphasize, in my own recent experience with Cryptococcus and sequencing 3,000 or 4,000 genomes, and now looking back at these things, I'm becoming a believer that people are infected with more than one strain. And so I think that's an emphasis that you put up there, and I think we need to pay attention to that. For you, I have a question that was always kind of hard for me. The Aztec study. Well, okay, it looks good. NIH paid, I don't know how much money for 6 or 7 years of collecting it, and you have one of the great biorepositories of Aspergillus of any place.
Hundreds of thousands.
But the other day, I got a call, like, Dr. Perfect, I think you need to get another job to pay for that Aztec thing. What do I mean? Dr. Wingard is retiring. The freezers are sitting there. He's not going to leave them anymore. And Dr. Alexander comes in my office and says, I think I have to take them or all of them are going to be dumped down the drain. So you got to come up with money to get them here for me while I try to figure out how to get money to keep them going. Okay, so the point of these things is biorepositories. I mean, NIH didn't think past this thing. These things are very important, all the data on that stuff. And who is minding the ship that's going to keep these things going for times that people can access them? You mentioned that challenge. It's right there. It's right there. Did anybody want to pay for the Aztec thing to keep it going? That's what we're dealing with right now. So I do want to emphasize to you that the challenge a lot of times is to have the freezers and have them paid attention to, have somebody watching them, and that's not necessarily cheap. The next question I want to do is, Mahmoud, you talked about a whole bunch of microbiomes and all this stuff with using various types of products. And do you think there's any impact of our diet? I mean, you wrote a diet, I say you never talked about diet. I got a lot of data that shows diet's actually pretty important, and pretty important to antifungal activity. So you kind of didn't put it out there, kind of interesting, but I would suggest that there are trickiness of these things, not just probiotics, but the things we eat may impact how we effectively treat fungal infections.
So basically, you opened for me a chance to advertise.
I'm sorry.
I wrote a book called Total Gut Balance, which is completely about microbiome, the science behind it, and then suggested a diet with even nice recipes, I believe, or at least what I got feedback, where definitely diet is a very, very important aspect of the managing your microbiome. And when I talk about nutritional supplements, it's a supplement. It's not the key thing to use, you know. So, but you are right. Diet and lifestyle, stress, exercise, and
some of his diets and used fluconazole. You can make fluconazole a really powerful drug, a fungicidal drug. Sometime I'll tell you about that. And finally, for Pete, yeah, you put a lot of things up there we do. One of the things I would like to emphasize that I think we need to think about is not only these biorepositories, but get the kind of patients that we need. One of the things is people have all this data and databases and stuff like that. But what we haven't captured very well, that's what Melissa did very well, and I wish we had a whole mess of these things, is the genetics around this thing, genetic susceptibility. You talked about one case at a time, but it'd be so much better if we also had lots of patients so that you can tweak some of these issues. So I would encourage us to think about the ability to have biorepositories or specimens, and not just on the organism side. I mean, we've got thousands of genomes now being sequenced, but also on the host side, because it takes extra effort to get that informed consent for that DNA. We need to think about that, because that's going to be the future of making all these things, predicting who's who and what kind of groups you go into and what kind of risk factors you have.
I mean, I've been an advocate. Many of you know this. I think we should approach each patient we approach for a clinical trial to try and obtain a DNA sample as well. Getting that consent can be tough, but whether they consent or not, if they have a certain disorder, I mean, in our case, it's a lot of patients with candidemia, a lot of patients with crypto, et cetera. I think we need to have DNA samples, and then who runs that? Probably the fairest place to send that, where access to the specimens is pretty universal, I'm guessing, would be somebody in Leonicas's lab or somebody like that. But I think that's one of the points I was trying to make. We have an enormous power as a group to do this, especially for rare fungi that no one can do as a single institution.
Tom?
Thank you all for great presentations. And though there are many questions, for the sake of time, I'll focus one question on Jerry. Jerry, thank you again. Outstanding presentation. I would ask that we hearken back to the approval for isavuconazole for mucormycosis. It was a landmark regulatory and scientific achievement. It was achieved in part by the critical nature of control. What were the proper controls? And fortunately, we had access to Fungoscope, which was a well-constructed database, numbers were small, and then literature review. However, in an era of data, in an era of massive numbers potentially in electronic databases now, even though the granularity may be limited, how can we access this much larger group of patients with rare molds in particular, perhaps rare yeasts as well, but for example, diseases, mucormycosis, lamentisporiosis, scedosporiosis, scapulariopsis, for example, where we have antifungal agents that are now targeted for these diseases and for which there may be a potential regulatory pathway. But the critical question arises, to what do you control them? And if beyond the registries, which are either in their nascent status or established, but still relatively small in numbers, how can we access the large databases through these several major efforts that you've accomplished?
So again, if it's a pharmaceutical question, I mean, so I have my list of databases. Someone comes into my office and they pose a question, and we start ticking off the questions. You know, what's the most important part? Is it the diagnosis? Is it the medication? You know, what do we need to really need the granularity on? So if it's a medication question, really it narrows down the sources to two. You know, a data source like Vizient or Premier, which again is academic institutions, over 500 of them, you know, detailed inpatient data, diagnoses, to a certain extent labs, and the medications. If we're interested in the use of the drug not only in the inpatient setting but in the outpatient setting, and you want to trace its use during the inpatient encounter and then how the patient progresses once they're discharged, now the only thing on the table is something like Optum and MarketScan. In any given year, the MarketScan data has about 3 billion prescription records. So even for a drug that's prescribed, as I showed you with Pete's study, you know, they had a small number of institutions and, you know, garnered, you know, several hundreds. You know, with about 15 minutes worth of work, you know, I was able to show that the MarketScan data produced, you know, sometimes 10 times as many prescription records for that. So it's really a matter of just taking the question, matching it to the database, and then acknowledging what the limitations of it are. As I said, this is not meant to replace clinical trials. It's really meant to supplement them. And as we know, you see how things are supposed to work in a clinical trial, and then you see how they actually work by looking at them in real-world settings such as this. So it's just a marriage of the question with the data source, and it's the advantage of having, you know, a group like the MSGERC who has access to the whole range of data sources. If you're a hammer, everything looks like a nail. If you only have the national inpatient sample, that's the limit of your questions. Whereas if you have this whole buffet of databases, now you have, you know, you can answer the questions in a very much more targeted way.
Thank you.
Dr. Rex.
John Rexon, I'd like to build on what Tom said. I've been musing all day long on something that's bothered me for quite some time. Pete, this is the comment that I told you I wanted to make with you earlier. If this group wants to move the needle, it's our responsibility to know what our stakeholders want, not necessarily what we think is the right academic approach. It may be an academic approach, but really what do our stakeholders want? So let's take large data, real-world data, and real-world evidence. There is now, the FDA just released a draft guideline on the use of real-world data and real-world evidence. Do you guys know it? Do you know what's in it? If you don't, you're not going to succeed with moving the needle. Earlier today, Tom asked, or somebody asked, why isn't galactomannan used as a marker in invasive aspergillosis? Well, actually, there is a reason. Now, you may disagree with the reason, but if you don't know the reason, no amount of ranting and railing about saying it to the FDA louder won't change their mind. Which doesn't work with teenagers either, right? So in order to actually change the way that we develop data, change the way that regulators and payers see data, you actually have to start seeing the world through their eyes. And those of you who know my journey, you know, I went from being an academic to moving into industry, and I spent, in my first few years in industry, a lot of what I did was apologize to my new industry colleagues for things that I now realized had been untutored comments that were made from I actually wanted to be helpful, but I didn't really understand what was going on. And the title of this really would be, why did they do that? Why did the company, Jack Sobel, you asked, why did the company not use a fluconazole comparator with it? There was a reason. You may not like it, but there was a reason. And so I'm gonna add to Pete's list, which is that if the MSGERC is going to move the needle in this space and be seen as a thinking leading group, it's incumbent upon this group to understand the regulatory and payer perspectives. We have to see through their eyes in order to generate the kind of data that actually move the needle. And I think you could add that to your list because that's actually training everybody, and it builds on, what kind of statistical support do you need? All this gets linked together. So it's a really interesting conversation that you spun up here, and it's going to take some untangling. But until you, the academic community, know the answer to why did they do that study that way, you're not able to move the needle as effectively. So it's not so much as a question; it's kind of a challenge to the group, and let's start thinking about that issue. We've got people who've actually done, Kieran Marr has now got some experience with banging away through diagnostics. Mahmoud has some experience with, building a little company. You guys all have things you could teach that would change the way people look at how you develop studies and what kind of data you develop. So.
Thank you. Those are great insights, John.
Dr. Cantoyanes.
One idea potentially to add to your list, Pete, is there is a lot of interest, at least in academia or other payers, about the influence of changing climate or geoclimatic factors to fungal disease or virulence. Actually, there is a CDC provisionally approved grant involved about coccidiomycosis in West United States and fires and temperature. So I think it's a very complicated area, and also the COVID pandemic in India tended to suggest also there are some geoclimatic factors to be explored. So I think for a young person who likes big data and mining data, how the climate changes incidence of disease and severity and presentation is a novel idea out there. Second, to Tom Walsh and John Rex's question, I'm kind of reflected on my own experience. At MD Anderson, we have 200 patients, real-life patients with fusariosis that I've been collecting since I started there. 200 patients with mucormycosis, real-life patients, patients that 80% of them will never enter these artificial clinical studies. You see, because they have a refractory disease, they are heavily involved, they have co-infections, and those are very rich databases to contend. And I would offer, provided there is some regulatory impediments, to make those available for rare infections. We have 200 patients with fusariosis, and what we found, which is sometimes I'm holding my tongue not to make some comments like that, is for fusariosis, nothing has changed for 20 years of antifungals unless leukemia goes to remission, which is kind of a known factor. The same for mucormycosis to a lot of degree. And the third point I want to make to Mahmoud is we struggle with our own microbiome studies. We have this prospective gap study in leukemia that we collect, all this data, and the diet comment is correct. But I'm not an expert in the field, but some people question all these correlative studies with microbiome, and they think that metagenomic correlations are going to tell us the truth of the true interactions. And what is the comment about just sequencing ITS sequences or 28? Is this the whole story of microbiome, or you need metagenomic or metabolomic studies?
I think this is a very, very important question. The issue is with metagenomics is, as you know, it's a hot new area. Everybody wants to jump on that bandwagon. But if you look at the reviews that talking about it, even though you get more data from the host genetics and whatever, which really will be very helpful, but our understanding to do the bioinformatics of it is really very, very complicated. And I would say in 5 years, once we refine this, and it will be much, much better. But at the moment, we have such a long experience with the 16S and ITS that we are able, and especially when we have thousands of people who submitted their fecal samples, we are able to look at what is a control. Till now, people in the microbiome area, they don't agree what is a control because it started with 250 people at the Human Microbiome Project. Now we have thousands of people where we use the same inclusion-exclusion criteria to try to separate people to say these are what we call control, healthy control. And this is helping us a lot in looking at different people personalization as well as different cohorts and find what is the dysbiosis. So that's where I think at the moment We have a wealth of information using 16S and ITS. At the same time, you bring something very, very important, which I really, because of the time limit, I did not talk about, is metabolomics. I think this is the next hot thing. It's like, you know, before I was, when my biofilm work started, okay, we did so much, then I start thinking, oh, maybe the microbiome is a good area. And now we've been over 10 years in it. Guess what? The next hot thing is the metabolome and also the postbiotics, which we started to collect data on that.
Thank you very much. We're at time, unfortunately, Ashraf, so I'm going to ask you to approach the podium after we're finished. Thank you very much for this incredible session.